Welcome to Nutrigenomics. Throughout your graduate program in nutritional sciences thus far, you've learned a lot about the metabolic, physiological, and behavioral basis of nutrition. At the heart of each of these, even behavior, are the action and influence of genes. Through your core course in the molecular aspects of nutrition, you already have a good foundation for gene structure and expression. This course will provide additional context for the use of genetic information in nutrition and dietetics, as well as expose you to a number of useful tools for identifying and evaluating the potential effects of genes and genetic variation. Nutrigenomics provides a multivariate picture of the many ways in which nutrition can influence health. In this context, whole human assessment can be performed at many different levels, including the level of phenomics, which is really just a fancy term for characterizing all the many traits and behaviors associated with nutrition. It can be measured at the level of genomics, which includes both single genes and the variation across the human genome. It can be measured at the level of proteomics, which is a technique for characterizing all the proteins in a body at a given time. It can be measured at the level of metabolomics, which is a comprehensive methodology for characterizing metabolites and metabolism. And it can be measured at the level of the microbiome. In terms of nutrition, the gut and oral microbiomes are probably the most important. This course is designed to build upon all that you've learned to date within the framework of genetics and a whole host of other measures to provide you the foundation for understanding how personalized or precision nutrition recommendations are made. So which organism do you think has the most genes? On the left is one of the simplest and most studied organisms in biology, C. elegans, or the lowly nematode, as it's also known. On the right is one of the most complex organisms on the planet in terms of physiology, behavior, and cognition, Homo sapiens. Scientists estimate the number of genes in C. elegans to be about 20,000. And believe it or not, the latest estimate in the number of genes in the human genome is just over 21,000. So why are humans so much more complex than worms? We can use an analogy from nutrition to explain this phenomenon. If I take a simple egg, I can combine it with any number of different ingredients. I can prepare or cook those different ingredients using different utensils at different temperatures and with different techniques. And I can create a whole host of different products at the end. How those products taste will be a function of the quality and quantity of each of the ingredients, down to what kind of feed was given to the chickens that laid the egg. In the same manner, the product of a single gene can interact with many other genes, each with the potential to be variable. Those genes can be regulated in any number of ways, including chemical modifications induced by or interacting with environmental exposures to ultimately produce the phenotype or state of the person. Since all humans, no matter how genetically similar, are unique, it stands to reason that individuals may require specific dietary recommendations to optimize health. Diets can be personalized based on specific traits or diseases, microbial abundance, metabolic response, current nutritional status, or physical activity level, and all of these conditions are influenced by genes. So how is the course structured? There are 12 content modules with 2 modules presented each week. In Module 1, we'll talk about inheritance and how traits are passed on from parents to children. In Module 2, we'll talk about genomic structure, some of which you've already covered in your molecular nutrition course, so this will be a bit of a review. In Module 3, I'll show you some genomics technologies, how these variation in the genome is actually measured. In Module 4, I'll provide you with some bioinformatics tools, some of which are web-based, so that you can use them to find out more about gene structure and sequence variation. In Module 5, we'll talk about nutritional biomarkers and how they're identified and measured. In Module 6, we'll do a little bit of genetic analysis, and some of that will be review from your statistics course. In Module 7, we'll talk about neural sensing and the complex networks in your brain that regulate feeding behavior. In Module 8, we'll talk about nutrient metabolism and how genes and diets interact to influence metabolism. In Module 9, we'll talk about gene regulation at the level of epigenetics and the microbiome. In Module 10, we'll talk about timing and circadian rhythms. In Module 11, we'll talk about the process of creating a personalized diet. And in Module 12, we'll actually apply those personalized diets in different settings. Each module is structured in a similar manner, with a series of lessons containing materials to read, watch, explore, and analyze. Short checks for understanding are given at the end of each lesson. Each module will contain a key concept or question for discussion, and each module will include a module project that can range from creating a family pedigree to applying the use of genomics tools to understand a personalized diet prescription. Throughout this course, examples from nutrition are interwoven into the information about genes and molecular function. Each module is worth a total of 100 points, with 25 points for all checks for understanding within a module, 25 points for each discussion, and 50 points for each module project, for a total of 1,200 points for the course. To make an A, you need at least 1,080 points. To make a B, you need 960 points. And to make a C, you need at least 840 points. Mastery of a topic is sometimes achieved through additional learning and application of the materials in different contexts. So the final exam is optional and designed to provide an opportunity for revisiting the materials learned in each module to make up points missed during the course. The final exam will consist of 12 questions, each associated with a specific module. Each question is worth up to 25 points, which can only be applied to the associated module, for a total of 100 points per module, or 1,200 points for the course. In other words, if you missed points in modules 2, 3, 4, 5, 7, 8, 10, and 11, as in this example, you can answer the questions associated with each of those modules to add up to 25 points for each module score. If you've made your target grade at the end of the 6 weeks, you do not have to take the final exam. Understanding personalized nutrition is essential for every clinician or nutrition specialist. By the end of this course, you will have the tools and knowledge to talk with patients and clients all about aspects of personalized nutrition. You are the nutrition professional of the future.